Hello, I'm Ayse Dincer. I'm doing my PhD in computer science at the University of Washington. Today, I'm going to provide an overview of my recent and ongoing project where we infer peptide coefficients from quantitative mass spectrometry data. Tandem mass spectrometry, or MSMS, can be used to quantify thousands of peptides in a complex biological mixture. However, the problem is that these measurements depend in part on the properties of the peptide sequence. Our goal is to train a machine learning model to quantify these peptide-specific properties, and then adjust the observed quantities to remove these effects. And our end goal is to offer our approach as a pre-processing step to eliminate peptide bias from quantitative MSMS data. Our approach rests on two primary assumptions. The first hypothesis is that peptide measurements can be decomposed into subcomponents. Here we have a schematic of our matrix Q of peptide measurements, and we denote one cell of these metrics as QIK, the observed abundance of peptide I in run K. Our hypothesis 1 states that QIK can be decomposed into a peptide coefficient Ci and an adjusted abundance alpha Ik. We then build our second assumption on the first one and state that unique sibling peptides should have equal adjusted abundances within a single MSMS run. By unique sibling peptides, what we mean is the peptides that occur exactly once in the protein database and that co-occur on a given protein sequence. So hypothesis 2 states that for run K and for sibling peptides I and J, alpha Ik should be equal to alpha Jk, which are the adjusted abundances. We use these two assumptions to infer peptide coefficients from peptide measurements to reveal the adjusted abundances. Accordingly, we implement a deep neural network to predict peptide coefficients from the amino acid sequences of peptides. And we then demonstrate the ability of this neural network to predict coefficients for new runs and new peptides. For our experiments, we analyzed quantitative MSMS data from human cerebrospinal fluids collected by data-independent acquisition. We also eliminated all shared peptides, the peptides that appear in more than one protein, before training our model. Our loss function for our neural network is based on the key assumption that the adjusted abundances of sibling peptides should be equal. So we are trying to minimize the distance between adjusted abundances of all pairs of sibling peptides I and J in our dataset. And we naturally calculate this loss over all runs and proteins. We also have some regularization losses to ensure proper scaling of the coefficients. And we then define our neural network to minimize this loss. We define a coefficient predictor network that takes as input a one hot encoded peptide sequence and produces as output a coefficient Di. We then parameterize a deep neural network model with a Siamese architecture. So this means that we have two copies of this coefficient predictor network, and these two networks have the exact same architecture and weights. And each arm of this network takes as input a peptide sequence and outputs a coefficient. And we train this network using all pairs of sibling peptides I and J to minimize our loss function. And one key point is that even though we train this model from only the unique peptides, it can also be used to make predictions for shared peptides. We showed that our neural network can predict coefficients that are generalizable across peptides and runs. For this experiment, we first split our peptides and runs into train and test sets randomly. Here you see the distribution of observed abundance ratios for the trained peptides and runs. So in other words, for all pairs of sibling peptides Pi and Pj, we're plotting Qik divided by Qjk. We then adjusted the abundances in this plot using the coefficients we inferred, di and dj. So this time you see the distribution of adjusted abundance ratios. And when we compare the two distributions, we can see that we reduce the variance in the distribution and also reduce the loss, where the loss is the log squared distance between sibling peptide abundances. But the more interesting aspect is that when we created the same plots for the test peptides, we could again observe that we reduce the loss and make the distribution of adjusted abundances more peaked at 1, which indicates that many pairs of sibling peptides are having closer abundances. So this is a promising result, indicating that our neural network can make accurate predictions for new sets of peptides and runs. Looking ahead, we want to investigate transferring our model across datasets to show that our model is generalizable across datasets as well. And we also want to explore the extent which our approach can improve the accuracy of MSMS measurements and can help with various quantitative MSMS analysis. Thank you for listening.